The scene opens with a comprehensive geological timeline, spanning Earth's history. The timeline focuses on the Cenozoic era, highlighting its major epochs. The Neogene period is further emphasized, pinpointing the Pliocene epoch. A marker precisely indicates 5.1 million years ago within the Pliocene epoch. The visual representation provides context for understanding Earth's deep history and the placement of specific events. The timeline focuses on a map, highlighting the present-day location of Chad in Africa. The Sahelanthropus fossils were discovered in Chad. Chad's location is crucial to understanding the context of the Sahelanthropus discovery. The discovery site provides insights into early hominid evolution. The map serves as a visual anchor for understanding the geographical context of the fossil find. Paleo-environment reconstruction visually represents the Lake Chad Basin as it existed 5.1 million years ago. The ancient Lake Chad Basin consisted of a mosaic of diverse environments. Woodlands formed a significant part of the Lake Chad Basin's paleo-environment. Grasslands and lakes completed the environmental mosaic of the Lake Chad Basin. Climate simulations project a future Chad experiencing warmer temperatures and increased precipitation. Higher rainfall and humidity levels are key indicators of the simulated climate shift. The simulation models the transition from current arid conditions to a wetter, more temperate environment. These climate models provide insights into potential environmental changes and their impacts. The Sahel region features diverse vegetation, including various tree species adapted to arid conditions. Grasses form a significant component of the Sahel's flora, providing ground cover and supporting local ecosystems. Shrubs are common in the Sahel, offering resilience against harsh environmental conditions. The vegetation of the Sahel is characterized by its adaptation to limited water availability. Flora in the Sahel plays a crucial role in preventing soil erosion and maintaining ecological balance. Different plant species in the Sahel exhibit unique survival strategies to thrive in the arid climate. Sahelanthropus tchadensis coexisted with various animal species in the African savanna. The fauna included early forms of elephants, adapted to the savanna environment. Giraffes were among the other mammals that shared the habitat with Sahelanthropus. These animal species were typical of the African savanna during the time of Sahelanthropus. The savanna environment provided a diverse range of resources for both hominids and animals. Understanding the associated fauna provides context for the ecological niche of Sahelanthropus. Sahelanthropus inhabited a transition zone between woodland and grassland environments. This habitat provided a mix of resources, including shelter in the woodlands and foraging opportunities in the grasslands. The woodland-grassland interface presented both opportunities and challenges for early hominids. The varied terrain offered diverse food sources, influencing the diet and survival strategies of Sahelanthropus. The open grasslands necessitated adaptations for predator avoidance and long-distance visibility. The Taurus Manala fossil site is a significant location for understanding early hominin evolution. The discovery of the Tumai skull at Taurus Manala provided crucial insights into early hominin species. Fossil discoveries at Taurus Manala contribute to the broader understanding of human origins and evolutionary pathways. The Taurus Manala site offers a window into the environment and conditions in which early hominins lived. Ongoing research at Taurus Manala continues to reveal new information about early human ancestors. The Tumai skull exhibits distinct anatomical features that provide insights into its evolutionary placement. A notable characteristic of the Tumai skull is its relatively small brain size. The prominent brow ridge is another significant anatomical feature of the Tumai skull. These anatomical features contribute to understanding the evolutionary position of Tumai. Detailed anatomical analysis of the Tumai skull provides crucial data for paleoanthropological research. Analysis of the Sahelanthropus femur provides evidence supporting bipedalism. Femoral neck angle is a key indicator. A more extended angle suggests upright posture. The distribution of cortical bone thickness in the femur reflects weight-bearing patterns associated with bipedalism. Anterior curvature of the femur shaft is consistent with bipedal locomotion. The morphology of the femoral condyles influences knee joint stability during bipedal walking. Comparative analysis with other hominids and primates aids in confirming bipedal adaptations in Sahelanthropus. Dietary reconstruction of Sahelanthropus suggests a predominantly plant-based diet. The primary food sources for Sahelanthropus were likely plants, providing essential nutrients and energy. Occasional meat consumption supplemented the plant-based diet of Sahelanthropus. The dietary habits of Sahelanthropus reflect an adaptation to available food resources in their environment. Sahelanthropus faced environmental challenges, including competition for limited resources. Competition for resources involved interactions with other animal species sharing the same habitat. Limited access to water and food sources increased the struggle for survival. The environment presented constant challenges to Sahelanthropus's survival. Sahelanthropus tchadensis may have faced threats from early forms of large carnivorous predators. Potential predators included early ancestors of modern big cats, adapted for hunting in open environments. Other carnivorous animals also posed a significant threat to early hominids. The vulnerability of Sahelanthropus was heightened by its relatively small size and lack of advanced weaponry. Fossil evidence suggests that interactions between early hominids and predators were common and often resulted in fatalities. Climate variability involves seasonal environmental changes. Resource availability fluctuates throughout the year due to seasonal shifts. Seasonal changes impact the distribution and abundance of flora. Fauna adapt their behavior and migration patterns based on seasonal resource availability. Seasonal changes influence water cycles, affecting water resource management. Understanding seasonal climate variability is crucial for predicting environmental changes. Water scarcity presents significant challenges during the dry season. Sahelanthropus may have adapted to locate scarce water sources. Access to water is crucial for survival during prolonged drought conditions. Survival strategies include digging for groundwater and following animal migration patterns. Sahelanthropus groups likely developed social structures to enhance survival. Potential group dynamics may have included cooperative hunting strategies. Social structures could have facilitated resource sharing and protection from predators. Visualization suggests potential social hierarchies and roles within Sahelanthropus groups. Cooperation within social structures may have improved overall group fitness and resilience. Early hominids, such as Sahelanthropus, may have utilized simple stone tools for basic tasks. These tools could have been used for foraging, such as breaking open nuts or digging for roots. Stone tools might also have provided a means of defense against predators. The use of tools represents an early stage in technological development and adaptation. Sahelanthropus may have developed behavioral adaptations to survive environmental changes. Behavioral adaptations could include changes in foraging strategies to find food in scarce environments. Altered social structures may have aided survival, such as forming larger groups for protection. Migration patterns could have shifted in response to seasonal resource availability. These behavioral shifts represent crucial adaptations for survival in a fluctuating environment. Gradual environmental shifts can significantly alter habitats over extended periods. These changes include variations in landscape, climate, and availability of resources. Simulations can illustrate the potential habitat changes during the lifespan of Sahelanthropus. Deforestation, desertification, and altered water cycles can reshape ecosystems. These environmental shifts can impact the survival and adaptation of species. Understanding these changes provides insights into the environmental pressures faced by early hominids. Sahelanthropus tchadensis is recognized as one of the earliest known species in the hominin lineage. Its discovery provides crucial insights into the initial stages of human evolutionary history. The position of Sahelanthropus helps to clarify the evolutionary relationships among different hominin species. Studying Sahelanthropus contributes to a deeper understanding of the traits and adaptations that define early hominins. Comparative anatomy reveals key evolutionary changes between Sahelanthropus and modern humans. Cranial capacity differences are evident in the smaller brain case of Sahelanthropus compared to modern humans. Facial structure exhibits significant variation, with Sahelanthropus possessing a more sloping face. Dental arcades differ, with Sahelanthropus showing a more U-shaped dental arrangement. Postcranial elements suggest differences in bipedal locomotion capabilities. Sahelanthropus exhibits a relatively small brain size compared to later hominin species. Later hominin species demonstrate a gradual increase in brain capacity over evolutionary time. The increase in brain size reflects the development of more complex cognitive functions. Relative brain capacity is a key indicator of hominin evolutionary advancement. Sahelanthropus exhibits a prominent brow ridge, a key cranial feature for analysis. The facial structure of Sahelanthropus provides insights into its evolutionary position. Detailed examination of cranial features aids in understanding the evolutionary implications of Sahelanthropus. The robust brow ridge is a distinguishing characteristic of Sahelanthropus's cranial morphology. Analysis of the facial structure reveals evolutionary relationships between Sahelanthropus and other hominids. Dental morphology analysis provides insights into the diet and feeding habits of species. Sahelanthropus dental morphology reveals specific dietary adaptations. Tooth structure, including enamel thickness and cusp patterns, correlates with food processing requirements. Analyzing dental morphology helps reconstruct the paleoecology and dietary niche of extinct hominids. Postcranial anatomy includes the skeletal elements located behind the cranium, primarily the limbs and torso. Limb proportions are crucial for understanding locomotion methods, such as bipedalism versus quadrupedalism. The relative length and robustness of limbs can indicate whether an organism primarily walked on 2 legs or 4. Habitat use, such as arboreal or terrestrial environments, influences the evolution of limb proportions. Analysis of Sahelanthropus's postcranial anatomy provides insights into its locomotion and potential habitat. Environmental pressures can drive the selection of advantageous traits in species. Sahelanthropus may have evolved specific traits that provided a survival advantage in their environment. These advantageous traits contributed to the evolutionary success of Sahelanthropus. Natural selection favors traits that increase an organism's ability to survive and reproduce in a specific environment. Fossilization is the process by which the remains of an organism are preserved in rock. The process often begins with rapid burial of the organism's remains under sediment. Over time, minerals replace the organic material, turning the remains into stone. Geological processes can then expose the fossilized remains through erosion or uplift. The fossilized remains of Sahelanthropus were preserved through this process over millions of years. Radiometric dating techniques are employed to determine the age of Sahelanthropus fossils. These methods rely on the decay rates of radioactive isotopes within the fossil or surrounding geological strata. By measuring the ratio of parent to daughter isotopes, the time elapsed since the fossil's formation can be estimated. Different radiometric methods are suitable for different time ranges, depending on the half-life of the isotopes used. The fossil site's geological context is revealed through stratigraphy. Fossils are embedded within distinct layers of rock and sediment. Stratigraphy helps determine the relative age of fossils based on their position within the rock layers. Each layer represents a different period in geological history. The composition and characteristics of each layer provide clues about the environment at the time of deposition. Analyzing the stratigraphy of a fossil site is crucial for understanding the history of life on Earth. Paleomagnetic analysis is used to determine the age of rock layers. This method relies on Earth's magnetic field recorded in rocks when they formed. Magnetic minerals in rocks align with Earth's magnetic field at the time of their formation. The direction and intensity of the magnetic field are frozen into the rock as it solidifies. By analyzing the magnetic orientation, the rock's age and the position of the magnetic poles at that time can be inferred. This information helps to date the rock layers at fossil sites, providing a timeline for the fossils found within them. Taphonomy encompasses the processes affecting fossil preservation. Taphonomic analysis examines factors influencing the state of Sahelanthropus fossils. Preservation can be affected by environmental conditions, such as sediment type and water chemistry. Biological factors, including scavenging and microbial activity, also influence fossil integrity. Geological processes, like erosion and tectonic activity, can alter or destroy fossil remains. Fossil reconstruction involves piecing together fragmented remains to understand ancient life forms. The process begins with careful cleaning and preparation of each fossil fragment. Advanced imaging techniques, such as CT scanning, aid in visualizing the internal structure of fossils. Digital modeling allows researchers to manipulate and reconstruct fossils virtually. Physical reconstruction involves assembling the fragments based on anatomical knowledge and digital models. The reconstructed Sahelanthropus fossil provides valuable insights into early human evolution. Scientists interpret fossil evidence to reconstruct the life and environment of Sahelanthropus. Analysis of the skull's features provides insights into Sahelanthropus's physical characteristics and potential behaviors. Geological context and dating techniques help determine the age and environmental conditions during Sahelanthropus's existence. Comparative anatomy with other hominids reveals evolutionary relationships and unique adaptations of Sahelanthropus. Inferences about diet, locomotion, and social behavior are drawn from the combined fossil and environmental evidence. Sahelanthropus's position within the evolutionary tree illustrates its relationship to other hominin species. The evolutionary tree visually represents the hypothesized lineage and divergence of hominin species over time. Phylogenetic analysis and fossil evidence are used to determine the placement of Sahelanthropus on the tree of life. Understanding Sahelanthropus's placement aids in tracing the origins and development of human evolutionary traits. DNA analysis, if available, could provide insights into Sahelanthropus's genetic makeup. Comparative genomics could reveal Sahelanthropus's relationship to modern humans and other hominids. Genetic markers could indicate potential adaptations to its environment. Analysis of ancient DNA could reveal information about population structure and migration patterns. Insights into genetic diseases and predispositions may be gleaned from DNA analysis. Genetic data could provide clues about the timing and rate of human evolution. Comparative genomics involves analyzing the genomes of different species to identify similarities and differences. Comparing the genomes of Sahelanthropus and other hominin species reveals evolutionary relationships. Genetic similarities between hominins suggest a shared ancestry and conserved functional elements. Genetic differences between hominins drive unique adaptations and species-specific traits. Comparative analysis helps identify genes responsible for human-specific traits by contrasting hominin genomes. Understanding genomic divergence provides insights into the evolutionary history and adaptation of hominin species. Gene flow can occur through potential interbreeding between different species. Sahelanthropus may have potentially interbred with other hominin species. Interbreeding could result in the transfer of genetic material between populations. The extent of gene flow depends on the frequency and success of interbreeding events. Potential gene flow influences the genetic diversity and evolutionary trajectory of hominin species. Environmental change may have significantly influenced the evolution of hominin species. Sahelanthropus, an early hominin, potentially evolved in response to specific environmental pressures. Adaptation to changing environments could have driven key evolutionary traits in hominins. Environmental shifts may have created selective pressures, favoring hominins with advantageous traits. The interplay between environmental dynamics and hominin evolution is a complex, ongoing area of research. Climate oscillations involve cyclical patterns of alternating wet and dry periods. These climate cycles significantly impacted the environment inhabited by Sahelanthropus. Wet periods fostered resource abundance, while dry periods led to scarcity and environmental stress. Simulation models help understand the potential climate conditions experienced by early hominids. Habitat fragmentation leads to the isolation of populations. Sahelanthropus populations may have become isolated due to habitat fragmentation. Isolated populations experience reduced gene flow, potentially leading to genetic divergence. Genetic divergence in isolated Sahelanthropus populations could have contributed to species evolution. Adaptive radiation describes the diversification of a species into multiple forms to exploit different ecological niches. Environmental change is a primary driver of adaptive radiation, creating new opportunities and selective pressures. Hominin species diversified in response to environmental changes, leading to variations in physical traits and behaviors. Different hominin species adapted to exploit specific food sources, influencing their dental structures and foraging strategies. Adaptive radiation resulted in a complex hominin family tree, with multiple species coexisting and competing for resources. Extinction can be influenced by multiple factors, including competition for resources, Environmental change is a significant driver of species extinction. Sahelanthropus may have faced extinction due to competition with other hominin species. Habitat loss and climate shifts can drastically reduce population sizes, increasing extinction risk. The interplay of competition and environmental change can create a synergistic extinction pressure. Sahelanthropus tchadensis provides crucial insights into early hominin evolution. Its discovery challenges previous assumptions about the timing and location of human origins. Sahelanthropus exhibits a mosaic of primitive and derived traits, complicating linear evolutionary models. The fossil evidence of Sahelanthropus contributes to a more complex understanding of the hominin family tree. Ongoing research focuses on visualizing Sahelanthropus's life and environment. Advanced imaging techniques reconstruct Sahelanthropus's cranial structure and facial features. Paleo-environmental studies analyze fossil pollen and sediment layers to recreate the ancient environment. Computational models simulate Sahelanthropus's biomechanics and movement patterns. Comparative anatomical studies analyze similarities and differences between Sahelanthropus and other hominids. Technological advancements provide new tools for analyzing Sahelanthropus fossils. 3D scanning enables the creation of detailed digital models of Sahelanthropus fossils. Computational analysis allows for the reconstruction of damaged or incomplete Sahelanthropus fossils. Advanced imaging techniques reveal internal structures and hidden features within Sahelanthropus fossils. International collaborative research efforts focus on the study of Sahelanthropus. Scientists from around the world contribute diverse expertise to analyze Sahelanthropus fossils. Collaborative studies enhance understanding of Sahelanthropus's evolutionary significance. Shared research data accelerates the pace of discovery in paleoanthropology. Public engagement is crucial for disseminating scientific findings and fostering understanding. Communicating Sahelanthropus research to the public enhances awareness of human origins and evolution. Effective public communication involves simplifying complex scientific data without sacrificing accuracy. Public engagement fosters support for scientific research and promotes informed decision-making. Ethical fossil collection involves responsible stewardship of paleontological resources. Proper documentation and contextual data are crucial for understanding the scientific value of fossils. Fossil preservation ensures the long-term availability of specimens for research and education. Commercial exploitation of fossils can lead to habitat destruction and loss of scientific information. Ethical guidelines promote collaboration between collectors, researchers, and landowners to protect paleontological heritage. Conservation efforts focus on protecting fossil sites to preserve them for future study and appreciation. Protective measures include controlled excavation, documentation, and stabilization of discovered fossils. Fossil sites are vulnerable to natural erosion and human interference, necessitating active conservation strategies. Preservation ensures that future generations can study and learn from the paleontological record. Museum exhibits showcase Sahelanthropus fossils to educate the public. Sahelanthropus tchadensis is a hominin species dated to approximately 7 million years ago. The discovery of Sahelanthropus provides insights into early human evolution. Exhibits detail the anatomical characteristics of Sahelanthropus, such as its small brain size and brow ridge. Interactive displays allow visitors to explore the evolutionary relationships between Sahelanthropus and other hominins. Museums play a vital role in communicating scientific discoveries and fostering public understanding of human origins. Documentary films can effectively communicate scientific concepts to a broad audience. Visual storytelling enhances audience engagement with scientific discoveries. Documentaries bring the story of Sahelanthropus to life through compelling visuals and expert interviews. Reconstructions and animations provide a visual understanding of Sahelanthropus's environment and lifestyle. Expert commentary in documentaries contextualizes the scientific significance of Sahelanthropus. Documentary films inspire public interest and support for scientific research and exploration. Educational resources are used to teach about human evolution. Sahelanthropus plays a role in the narrative of human evolution. Visual aids enhance understanding of human evolutionary history. Interactive simulations provide an engaging learning experience about early hominids. Scientific publications are essential for disseminating research findings. Publications facilitate the broad sharing of data, methodologies, and conclusions. Peer review ensures the quality and validity of published research. Shared research findings contribute to the collective advancement of scientific understanding. Publications on human evolution enhance our knowledge of human origins and development. Open access publications promote wider accessibility and accelerate scientific progress. Peer review is a critical process for ensuring the scientific rigor of Sahelanthropus research. The process involves experts critically evaluating research methods, data, and interpretations. Peer review helps to identify potential errors, biases, and alternative explanations. This process enhances the credibility and reliability of published findings. Ultimately, peer review strengthens the foundation of knowledge about Sahelanthropus. Statistical methods are employed to analyze data derived from Sahelanthropus fossils. These methods help determine the age and characteristics of the fossils. Comparative analysis involves comparing Sahelanthropus fossils with those of other hominids. Multivariate analysis assesses multiple variables simultaneously to reveal relationships. Statistical significance testing validates the reliability of the results. Morphometrics involves the measurement and analysis of shapes, particularly in biological structures like bones. Geometric morphometrics uses coordinate data from anatomical landmarks to quantify shape variation. These measurements are used to statistically analyze shape differences between different groups or species. Principal component analysis, PCA, is used to reduce the dimensionality of shape data and identify the major axes of variation. Deformation grids visualize shape differences by showing how one shape is transformed into another. Morphometric analysis provides insights into the evolutionary relationships and functional adaptations of extinct hominins. Finite element analysis, FEA, is a computational method used to simulate the biomechanical properties of materials. Phi divides a complex object into smaller, simpler elements to analyze how each element responds to applied forces. The simulation calculates stress, strain, and displacement within each element, providing a comprehensive understanding of the object's behavior. FEA allows researchers to predict how Sahelanthropus bones would respond to different loading conditions. By simulating biomechanical properties, FEA helps in understanding the functional adaptations of ancient hominin bones. FEA provides insights into the evolutionary pressures that shaped the skeletal structures of early hominins. Computed tomography, CT scanning, is utilized to generate three-dimensional models of fossils. CT scanning enables the visualization of internal structures within fossils without physical dissection. The process involves capturing multiple X-ray images from different angles to create a comprehensive dataset. Sahelanthropus fossils can be digitally reconstructed and analyzed using CT-generated 3D models. Microscopy techniques are employed to analyze the microscopic structures of Sahelanthropus bones. Microscopic analysis reveals detailed information about the bone's composition and formation. Microscopy is also used to examine the microscopic structures of Sahelanthropus teeth. Analyzing tooth microstructure provides insights into diet and environmental conditions. Isotope analysis is used to reconstruct the diet and environment of Sahelanthropus. Different isotopes in Sahelanthropus remains provide clues about their food sources and habitat. Carbon isotope ratios in teeth enamel indicate the proportion of C3 and C4 plants in the diet. Strontium isotope ratios in bones reflect the geological composition of the region inhabited by Sahelanthropus. Pollen analysis is a method used to reconstruct past vegetation. The process involves extracting and identifying pollen grains from soil samples. Different plant species produce pollen grains with unique morphological characteristics. By analyzing the pollen record, scientists can determine the types of plants that grew in a region at different times. Pollen analysis provides valuable insights into environmental changes and human impact on past ecosystems of the Sahel region. Charcoal analysis provides evidence of fire use by early hominins. Microscopic analysis of charcoal reveals cellular structures indicative of controlled burning. Radiocarbon dating of charcoal determines the age of the fire event. The presence of charcoal in archaeological layers indicates human activity and environmental modification. Charcoal analysis helps reconstruct past environments and human behaviors. Examining the spatial distribution of charcoal provides insights into hearth locations and fire management strategies. Sediment analysis is a crucial method for reconstructing past environments. Analysis of sediment composition reveals information about climate, vegetation, and geological conditions. At the Sahelanthropus fossil site, sediment analysis helps contextualize the fossil finds. Reconstructing past environments provides insights into the ecological context of early hominin evolution. Geochemical analysis is employed to trace the origins of fossils. The elemental composition of a fossil provides clues about its geological context. Isotopic dating techniques determine the age of fossils through radioactive decay analysis. Geochemical signatures can reveal the environmental conditions present during the fossil's formation. Remote sensing techniques are employed to map fossil sites and identify potential excavation areas. Satellite imagery and aerial photography are used to detect surface features indicative of fossil deposits. LiDAR, light detection and ranging, technology generates detailed three-dimensional models of terrain, revealing subtle topographic variations. Spectral imaging analyzes the composition of surface materials, identifying minerals associated with fossil preservation. Geophysical surveys, such as ground-penetrating radar, can detect subsurface anomalies indicative of fossil-bearing layers. Data integration from multiple remote sensing sources provides a comprehensive understanding of potential fossil sites. Geographic Information Systems, GIS, are used to manage spatial data. GIS enables the analysis of spatial data related to Sahelanthropus fossils. Visualization is a key component of GIS for understanding spatial patterns. GIS integrates diverse datasets to provide a comprehensive view of fossil locations. Spatial analysis in GIS aids in identifying potential new fossil sites. Virtual reality, VR, technology creates immersive experiences, allowing users to explore environments. VR enables the reconstruction and exploration of past environments, such as the habitat of Sahelanthropus. Users can interact with and study virtual representations of ancient hominids and their surroundings. VR simulations offer a unique perspective on the lives and environments of early hominids. The immersive nature of VR enhances understanding and engagement with historical and anthropological subjects. VR applications in paleontology provide new opportunities for research, education, and public outreach. Augmented reality, AR, overlays digital information onto the real world. AR enhances understanding by providing contextual data in real time. AR applications include the visualization of complex scientific data. AR systems use sensors and tracking technology to accurately align digital content with the physical environment. The overlay of digital information can improve the analysis and interpretation of Sahelanthropus fossils. AR provides interactive and immersive experiences for scientific exploration. 3D printing technology facilitates the creation of replicas of Sahelanthropus fossils. Digital scans of original fossils are used as blueprints for the 3D printing process. 3D printed fossil replicas serve educational purposes, providing accessible learning tools. These replicas also support research by allowing scientists to study fossil structures without risking damage to the original specimens. The use of 3D printing ensures accurate reproduction of complex fossil morphologies. Artificial intelligence is employed to analyze large datasets related to Sahelanthropus fossils. AI algorithms identify patterns and trends within the fossil data that may be imperceptible to human observation. The AI system processes diverse data types, including fossil morphology, geological context, and comparative genomics. Visualization tools enable researchers to interact with the AI's findings and explore the complex relationships within the data. Machine learning techniques are employed to predict potential fossil site locations. Geological data, climate patterns, and existing fossil records are key inputs for predictive models. Algorithms analyze complex datasets to identify correlations indicative of fossil presence. Predictive models output probability maps highlighting areas with high fossil discovery potential. Machine learning enhances the efficiency and accuracy of paleontological expeditions. Robotics technology is increasingly utilized to aid in the delicate process of fossil excavation. Robots can perform tasks that are too dangerous or precise for human paleontologists. Advanced imaging systems on robots allow for non-destructive analysis of fossils in situ. Robotic systems can map excavation sites with high accuracy, creating detailed 3D models. The use of robotics enhances the efficiency and accuracy of fossil discovery and documentation. Robotics facilitates the exploration of remote and challenging environments for fossil remains. Drones are utilized for aerial surveys of fossil sites, enabling comprehensive data collection. Aerial surveys facilitate the creation of high-resolution maps of fossil locations. These maps aid in the precise documentation and analysis of fossil distributions. Drone-based surveys offer a non-invasive method for monitoring and preserving paleontological resources. Satellite imagery provides a powerful tool for monitoring environmental changes across the globe. Deforestation patterns are tracked using satellite data to assess habitat loss and ecosystem degradation. Changes in polar ice caps are monitored to understand the impact of climate change on sea levels. Urban expansion and land use changes are analyzed to assess their impact on natural resources and biodiversity. Satellite monitoring helps to identify and assess the impact of pollution on ecosystems. Fossil sites are monitored using satellite imagery to protect them from environmental threats and human activities. The study of Sahelanthropus and other early hominin species is crucial to understanding human origins. The ongoing quest to understand our origins involves interdisciplinary research and collaboration. Fossil evidence provides direct insights into the physical characteristics and evolutionary adaptations of early hominins. Genetic analysis of ancient DNA offers valuable information about the relationships between different hominin species. Continued exploration and research are essential for unraveling the mysteries of human evolution. Please subscribe to the channel and hit the notification bell so that you will be updated for more videos.